Hi everyone, Dr. Mike here. In this video, we're taking a look at ketogenesis and diabetic ketoacidosis. Alright, to begin, we need to have a look at what happens in what we call the fed state. So we ingest a meal, that meal is going to have things like glucose in it, so let's pop that in. What we've got here is the bloodstream, so our systemic circulation. So we know that we've just had a meal and we've got increased glucose there in the bloodstream. We know that that glucose will ultimately travel to the pancreas, and the pancreas, very importantly, is going to sense that increase in blood glucose. Now remember, if I were to just very quickly draw up the pancreas, so there's our pancreas, there's going to be groups of cells called islet cells, and some of those cells will have what's called beta cells, some are going to be alpha cells, and when the high glucose reaches these beta cells, it stimulates them to release insulin. Now that's important because insulin will act upon the alpha cells and negatively affect them, inhibit them from releasing their hormone, which is called glucagon. So effectively, high blood glucose is going to simultaneously be floating through the bloodstream with high insulin. This is important because insulin is required for two major tissues of the body. Those tissues are muscle tissue, specifically skeletal muscle, and fat tissue, adipose tissue. Insulin tells those two tissues to bring glucose transporters to their surface and pull the glucose in from the bloodstream into the tissue. That's important because think about how much of your body mass is muscle and fat, a significant portion. So if insulin isn't present, a lot of that glucose is going to remain locked in your bloodstream. Now, here we've got the insulin. It's helped bring glucose into those two tissues. In addition to that, insulin is going to help trigger glucose, because glucose will enter the liver, right? We've got the liver here. We don't need insulin for glucose to enter the liver. But what insulin can do is it stimulates the process of glycolysis. Glycolysis is taking glucose and ultimately turning it into pyruvate. So what we've just highlighted here, by missing out on all of those important steps, is glycolysis. Now what insulin can also do is it stimulates pyruvate to enter the mitochondria. So what I might do is I'm going to draw up the mitochondria by drawing up that double membrane that it is. There we go. There's the double membrane of the mitochondria. Pyruvate will enter the mitochondria and it will turn into something called acetyl-CoA. Acetyl-CoA is the very first step in the Krebs cycle. Now, it needs to bind to something called oxaloacetate. So acetyl-CoA must bind with something called oxaloacetate, and then it starts to form, so it will form citrate and then a whole bunch of other different derivatives, and that's going to be the Krebs cycle. Now the whole point of the Krebs cycle is to spit out two really important molecules. These are NADH and FADH2. Now if you watch my electron transport chain video, or even the glycolysis video, or even the Krebs cycle video, you'll see that what these two molecules do is they simply hold on to electrons and protons. So they're holding on to electrons and they're holding on to protons. And that's important because they hand those electrons and protons to the electron transport chain embedded in the inner membrane of the mitochondria, right? They play hot potato with the electrons, they pump hydrogen ions, and the whole point of all this is to produce a huge amount of ATP. All right, high glucose ultimately produces a bunch of ATP. Brilliant. But what happens when our glucose levels drop? Our glucose levels are low. So now we're in a scenario here where, let's draw it over here, we've got low blood glucose. Well, if we've got low blood glucose, it will travel to the pancreas, but it's not going to stimulate those beta cells, right? It's going to stimulate the alpha cells. Now think about this. Low blood glucose is going to stimulate those alpha cells, and those alpha cells will release glucagon. Now remember, gluc, meaning glucose, gon, think about when glucose is gone, glucagon gets released. So now we're in a scenario of low blood glucose, which means we now have high glucagon. Now what glucagon can do is it can slow down glycolysis, or even try and inhibit glycolysis and inhibit the Krebs cycle. Its job is to try and replenish blood glucose. It's not thinking about energy at the moment. It just goes, remember, think homeostasis, right? We need to maintain our blood glucose levels at around about 5 millimoles per litre-ish, okay? So if it's going below that, we need to increase it. So glucagon goes, how can I increase blood glucose levels? I know, I've got an idea. I can take glycogen, which I've stored from occasions of I've eaten a lot of food and I've stored that glucose as glycogen. I can take that and just turn it into glucose. Brilliant. That glucose can then leave the bloodstream and help re-establish our blood glucose levels. Brilliant. What else could we do? Well, we could just reverse this whole process to produce glucose. That's a great idea. The problem is we can't take pyruvate and reverse it. Okay, so that's a bit of a problem. What can we do? I know what we can do. Glucagon can stimulate some other things. Glucagon can take things like triglycerides, so fat, right, here's our triglycerides, and stimulate it to break apart to form glycerol and fatty acids. So remember, triglycerides are made up of 3 fatty acids and a glycerol backbone. And it was glucagon that stimulated this process, a process known as lipolysis, stimulated by glucagon. And now we've got these glycerol and fatty acids in the bloodstream. What do we do with it? Well, no problem. Glycerol can turn into glucose, and glucose can leave and go back into the bloodstream. Brilliant, we're helping re-establish our blood glucose levels. This process of taking a non-carbohydrate based source and feeding it in to make more glucose is called gluconeogenesis. So when blood glucose levels are low, gluconeogenesis can occur. Okay, that's brilliant. What else can happen? Well, I want you to think about this. We can't bring pyruvate back up, but what we can do is take oxaloacetate. And oxaloacetate can leave the Krebs cycle and leave the mitochondria, and oxaloacetate can turn back into glucose. Now that's brilliant. But that also means that the levels of oxaloacetate in the mitochondria, in the Krebs cycle, drops down significantly. Pyruvate goes, hey, I know you, you're a friend of mine, I'm going to turn into oxaloacetate and turn into glucose. So while pyruvate can't go backwards immediately, it can turn into oxaloacetate and turn into glucose. So oxaloacetate is a gluconeogenic substrate. All right, this is good. What else can happen? Well, we can take amino acids that are floating through the bloodstream. Here's some amino acids. And we can take those amino acids and feed it into pyruvate. And pyruvate can turn into oxaloacetate, which can turn into glucose and re-establish blood glucose. So that means amino acids are a gluconeogenic substrate. So let's highlight some gluconeogenic substrates at the moment, right? We've got glycerol as a gluconeogenic substrate. Amino acids are a gluconeogenic substrate. Oxaloacetate is a gluconeogenic substrate. Another thing that's a gluconeogenic substrate is in our muscle tissue, lactate will have been produced, and that can turn to pyruvate. So that's a gluconeogenic substrate. All right, we've got glycerol, amino acids, lactate, oxaloacetate, but you're probably looking and going, wait a minute, what about the fatty acids? How do the fatty acids feed into this process? All right, well, fatty acids can't jump into pyruvate or this process going up to glucose, but what fatty acids can jump into is fatty acids can jump down, and we're going to have to do a little jump there and a jump there, and they turn into acetyl-CoA. Now, fatty acids, through the process of lipolysis, when blood glucose levels are low, fatty acids turn into acetyl-CoA, which means acetyl-CoA levels go up. But remember, oxaloacetate buggered off. It left to form glucose. But we need oxaloacetate to bind to acetyl-CoA. Now remember, this is happening when our blood glucose levels are low and our insulin levels are low, but our glucagon levels are high. So blood glucose levels low, insulin is low, glucagon is high. All right, let's think about what's happening. Not enough oxaloacetate to bind with acetyl-CoA. And so this then means that the acetyl-CoA accumulates. Now, if acetyl-CoA accumulates, they start snapping together like Lego blocks. Acetyl-CoA plus acetyl-CoA plus acetyl-CoA. We start snapping them together, and what we end up producing is something called ketones. Now, the first ketone that we produce is acetoacetate. And acetoacetate can split off and form acetone and beta hydroxybutyrate. Acetone and beta hydroxybutyrate formed from acetoacetate. Now, what they can do is they can leave the liver. Remember, all of this is happening in the liver. They can leave the liver, right, these ketones, and these ketones can travel to the brain and cross the blood-brain barrier. Oh, that's good. They can cross the blood-brain barrier. And now we've got ketones in the brain. And what can these ketones do? So let's just think. Our blood glucose levels were low. The brain only wants glucose for energy. If it doesn't have glucose for energy, it needs to figure out and find another resource. Luckily, the accumulation of these fatty acids formed acetyl-CoA, which snapped together to form the ketones, and they can leave. And ketones, because they're ultimately just made up of acetyl-CoAs, will turn back into acetyl-CoA, bind to the oxaloacetate, and then undergo the Krebs cycle. And what did the Krebs cycle produce? It produced the NADH and the FADH2. And I told you that those two things together, via the electron transport chain, produced ATP, which is what the brain's after. Now you might be looking at this going, wait a minute, Mike, you said that we didn't have enough oxaloacetate because it's jumping out. This only happens in the liver. The brain does not undergo gluconeogenesis. The brain does not try and re-establish blood glucose levels. So oxaloacetate levels are absolutely fine in the brain. And that's why the brain can use ketones as a backup energy source when our blood glucose levels are low. Now, this is the process of ketogenesis. But we haven't spoken about what happens in diabetes, because this is what happens when so this process of ketogenesis happens. Ketogenesis will happen when we have low blood glucose levels, low insulin levels, and high glucagon levels, which we've shown. Now, in a healthy individual, this generally just happens through carb restriction, right? Carbohydrate restriction, because that's a drop in blood glucose, which will drop your insulin, which will raise your glucagon. But there is a condition out there called diabetes mellitus. And in diabetes mellitus, we know that the individual, if it's type 1 diabetes, let's write this down here, if it's type 1 diabetes mellitus, that means that the insulin that person produces, well, there is no insulin that person produces, which is ticking that box there, right? And in type 2 diabetes mellitus, they do produce insulin, but the body really just ignores it, right? All right, so let's take type 1 diabetes, because this is the scenario in which the ketones can really get produced. Type 1 diabetes is an autoimmune disease. It generally happens earlier in life where the immune cells are attacking these beta cells of the pancreas. And if they've done that, there's no beta cells and there's no insulin. All right, these young individuals have no insulin in the body. Zero insulin. Now, what did I say before? I said that insulin stimulated glycolysis and the Krebs cycle to produce ATP. I also said insulin inhibited glucagon release. But if we've got no insulin, then there's no inhibition of glucagon. Now, they may not be restricting their carbohydrates. They may have normal levels of glucose in the bloodstream, but because there's no insulin, there's no inhibition of glucagon. So they paradoxically can have high glucose levels and high glucagon levels. And I said glucagon inhibited this process and stimulated lipolysis. So in people with type 1 diabetes, and they don't know they've got type 1 diabetes, they will have low insulin, high glucagon, and high blood glucose. And in the process, gluconeogenesis is happening to further increase the blood glucose, and the fatty acids through lipolysis are producing ketones. Now, what might be the problem here? Well, what might be the problem is the individual might get what's called DKA. Diabetic Ketoacidosis. And in order for somebody to be diagnosed with DKA, you actually need to tick the 3 criteria. The first of these criteria is the D, the diabetes side of things. They need to have high blood glucose levels, which we know is stereotypical of diabetes. If we don't know that they've got a diagnosis of diabetes, maybe they've got high blood glucose and a family history of diabetes mellitus. So they need to tick that box. The second box they need to tick is the K. Now the K is the ketones. So they need to have high blood ketones or high urinary ketones. So that's ticking that second box for the ketone production. And then finally, the A is something we haven't spoken about, which is acidosis. Now, in actual fact, it needs to be a high anion gap metabolic acidosis. All right, this is a lot of things. A high anion gap metabolic acidosis. What is this referring to? All right. When you, excuse me, when you snap these acetyl-CoA together and produce acetoacetate, and that acetoacetate snaps apart, the beta-hydroxybutyrate it produces, it actually, before it produces that, it forms beta-hydroxybutyric acid. Beta-hydroxybutyric acid. The definition of an acid is that it produces hydrogen ions and a conjugate base. The conjugate base is the beta-hydroxybutyrate. Remember, conjugate bases have negative charges associated with them, but the hydrogen ions are produced. Now, these hydrogen ions will jump into the bloodstream and accumulate. So now what do we have? We've got an acidosis. Now, not only do we have the acidosis, it's a metabolic acidosis because it's produced through metabolic pathways. If it's a respiratory acidosis, that's simply just saying we're accumulating carbon dioxide. Just so as a brief background, remember that carbon dioxide can bind with the water in your blood and produce H2CO3, which is called carbonic acid. That can split itself apart because acids hate themselves to produce hydrogen ions and bicarbonate ions. Accumulation of carbon dioxide can lead to an accumulation of hydrogen ions. That's called a respiratory acidosis. But here, the hydrogen ions are being produced through metabolic products, which are the ketones, and that's why it's called a metabolic acidosis. But then the question is, what is this high anion gap? All right. Anytime you produce acid, there is this thing called the anion gap that can be calculated. Now, the anion gap, so remember, you've got cations, which are positively charged things, and anions, which are negatively charged things. The anion gap takes cations like sodium and deducts the concentration of sodium and deducts from that the concentration of things like chloride added to bicarbonate. So here's the most common cation in the body, and here are some of the common anions, or most common anions in the body. Now, if we were to look at the normal anion gap, right? Sodium is going to be at around about 140 millimoles per litre, or milliequivalents, right? Chloride's going to be at around about 104, and bicarbonate's going to be around about 24. And if we have a look at this, and right, so add those two up, that's 128. 140 minus 128, what's that going to be? Around about 12, right? So the gap is around about 12 milliequivalents per litre, or millimoles per litre, right? That's the gap. All right, that's a normal anion gap. But what are we saying in this scenario with a high anion gap? We've just produced from beta-hydroxybutyric acid. It's split apart. Remember, that's an acid too. And it's split apart to the hydrogen ions and the conjugate base, which is negatively charged. So this splits off to form the hydrogen ions and the negative base called beta-hydroxybutyrate. When we release a whole bunch of hydrogen ions into the bloodstream, which we're doing here, the bicarbonate, this thing here, in our bloodstream goes, oh no, this isn't good. I need to bind to them and mop them up. So when it binds to it, bicarbonate disappears, and the product that it produces is H2CO3, which is this thing here, because this is reversible. The point I'm making is bicarbonate goes down. The concentration goes down because it's binding to that to form something else. Hopefully that makes sense. Which means this number is going to go down. All right. That number is going to go down. That makes sense. So let's now bring that down to 20 instead of 24. All right. So if we bring that down to 24, what do you think that then means? 140 minus 104 plus 20, so that's 124. 140 minus 124, 34, 4, 6, 16. So what has now happened is the gap has increased to about 16. Now, that's a high anion gap. The gap has gone up because we've lost bicarbonate. And we've lost the bicarbonate because it's bound to the hydrogen ions that have been produced. So now we've ticked the box here for the high anion gap. Now, what are the problems with this? Somebody's got type 1 diabetes, right? Type 1 diabetes. They don't have any insulin. Insulin's gone. So there's no negative regulation of glucagon. So we've got low insulin. Let's draw it up here, right? Now we've got this scenario. What's the scenario? The scenario is low insulin, which subsequently will mean high glucagon. They've still got carbohydrates, so they've got hyperglycemia. So they've got high blood glucose levels. And because the insulin's low, this process of glycolysis and Krebs cycle isn't being preferred. The high glucagon is stimulating glycogen to release more glucose, further increasing blood glucose levels. The glucagon is stimulating lipolysis, releasing glycerol, and gluconeogenesis, stimulating even more glucose in the bloodstream. So this person's blood glucose is going through the roof, ticking that box here for the diabetes side of things, right? And again, the fatty acids turning into acetyl-CoA, forming the ketones, forming the acid. They've got acidosis. What's the problem here? The problem is the acidosis. Because in the brain, we've got an area. We've got the midbrain, we've got the pons, and we've got the medulla, or the medulla oblongata. And at the dorsal end, we've got something called the chemoreceptive trigger zone. And the chemoreceptive trigger zone can be triggered by chemicals, right? Like hydrogen ions and ketones. And it will stimulate, it goes, oh, there's something in the blood that shouldn't be there. And what do you end up doing? Vomiting. Now, if you vomit, you can lose fluid in the body. So what can happen with DKA is vomiting, fluid loss because of the vomiting. Ultimately, if it remains, it can lead to a coma because of the acidosis. But before the coma, the acetone that's been released, can be breathed out, right? So if we take this acetone and bring it all the way out here, it can be breathed out via the lungs. And what do you think acetone smells like? Acetone smells sweet and fruity. And so you've got this sweet, fruity breath, which can happen because of the acetone that you've produced, breathing out via the lungs. So acetone doesn't necessarily contribute very much to acetyl-CoA production in the brain, but it is sweet and fruity. The other thing is that when you've got a lot of acid in the body, the brain goes, how do I get rid of this acid? Well, remember, that acid bound with the bicarbonate to form carbonic acid, this thing here. And what did I say carbonic acid can do? It can go backwards to form carbon dioxide. And what can we do with that carbon dioxide? Breathe it out. So another thing that can happen is hyperventilation. Hyperventilation, called Kussmaul breathing. So these are just some of the issues that can happen when somebody has diabetic ketoacidosis. Now, treatment for this, number one, is obviously going to be insulin, but also re-establishing fluid loss. And if the ion concentrations are out, which can happen, so very briefly there, when you've got heaps of hydrogen ions floating through the body, remember if you've got a cell, right? So here's a cell of the body, and it's filled with potassium, right? But if you're bathing that cell in hydrogen ions now, to save the body from the acidosis that's happening, because hydrogen ions can denature proteins, it tries to hide the hydrogen from the rest of the body. It throws hydrogen into the cells, but does it by exchanging it for potassium. So then our extracellular fluid has potassium in it. So our bloodstream now has more potassium in it. If you've got more potassium in the bloodstream, you pee out more potassium. In addition, so another thing I forgot, high blood glucose can lead to osmotic effects. So remember, glucose has a charge associated with it. It holds on to water. So if you, let's just say, here's the kidneys, right? Here's the kidneys. If you've got high glucose coming out in your pee, it's going to come along with a lot of water because it holds on to the water. And this is, firstly, it's called glucosuria, which is increased glucose in the urine, but also polyuria, which is you're peeing more frequently, but this also contributes to the fluid loss as well, right? But because now we've got more potassium in the bloodstream because we've exchanged it for the hydrogen ions, we also pee out more potassium. So we can become hypokalemic, or at least these individuals might become hypokalemic. And you might need to, when you re-establish fluids, re-establish potassium as well. All right, I think I might leave it there. This is a summary of ketogenesis and diabetic ketoacidosis. I'm Dr. Mike. Hi everyone, Dr. Mike here. If you enjoyed this video, please hit like and subscribe. We've got hundreds of others just like this. If you want to contact us, please do so on social media. We are on Instagram, Twitter, and TikTok at Dr. Mike Todorovic, at DrMiketodorovic. Speak to you soon.